Thank you. So, hello everyone. I would like to introduce what study metadata do we have available at the European Variation Archive and the diverse ways that people can consume it. So, the EVA is a relatively new resource here at EBI. So for those who don't know exactly what it is, I will try to explain it first thing during this presentation. And what are the benefits of archiving VCF files and consideration on correctness, metadata and data checks, etc. And then moving into what is available, I will present first the website, focusing mainly on the study metadata, and then on the programmatic access, the API that allows people to consume this information and integrate it in their own websites or with other tools, etc. And finally, future plans that we have in the context of BD2K and the Omics Discovery Index specifically. So what is the European Variation Archive? It is an open access repository that accepts all types of variation. We store short nucleotide variation, indels, structural variation, any type of variation is welcome. It is submission-based, so our growing depends on people submitting new studies. For the larger studies that were already existing when the EVA was began operating, we handled them. For instance, 1,000 genomes, UK10K, etc. Those we manage them directly, but ongoing work depends on submissions made by external contributors. We accept studies from any species also. So right now the majority of them are human-based, but we also have a lot of animal studies, plants, and others such as parasites. And all this data is available via the website or the API that I will describe later. So what are the benefits of archiving the VCF files? First is that we take very, very seriously the correctness of the metadata and the variant data. If there's no guarantee of the quality of this information, in practice this would be useless for later research. So during the submission process, we validate the metadata, we collaborate with the submitters to ensure that everything is correct and faithful to the research they've done. And we also validate all the variation data that comes in using our own automated tool that I'll mention later. Second, we broker all this information into other archives at EBI. So people doesn't need to submit, let's say, sample information to biosamples and then sequencing information to other parts. We handle it all together. Once the studies have been archived, we generate stable accessions, both for the studies themselves, for the files, and we also consume DB-SNP accessions for the variants. Once all this process is done, then the data can be shared from the single resource. Multiple studies can be integrated into a single site instead of having to visit, let's say, a 1,000 genomes portal, the exact portal, and having to consume them separately. They are all available at EVA, so it's just a one query and one-stop shop. And finally, we have a dynamic loading cycle. So instead of being tied to a, let's say, 3-month release cycle, we process the submissions as they come in. So they can be the accessions will be available in approximately 48 hours. Then the browsing on the website, we try to make it as quick as possible. So regarding how do we ensure the correctness of the VCF data. So after these 2 years operating, we have developed a validation tool because we detected that many of the VCF files that were already publicly available, even those from big studies, were not truly compliant to the specification. They had adapted some fields, and it was not possible to use them from other tools, or they needed some tweaking to be really useful for the community. So we implemented this tool that anybody can run from their own machine. We are also working with the Global Alliance for Genomics and Health to improve the specification, because we detected certain ambiguities that were causing people to make those mistakes. It wasn't that they wanted to do them voluntarily, but the spec wasn't clear about what the restrictions were in some cases. And as I mentioned earlier, we broker data to other repositories at EBI and outside EBI too. So the metadata and the files are also stored in the European Nucleotide Archive, which also allows to link the VCF analysis with the sequencing experiment in FASTQ or BAM format, for instance, which helps to have a better overview of what a study really covers. We also broker sample metadata to biosamples, short variants to dbSNP, structural variants to dbVar, and clinical relevant variants to ClinVar. So it's a lot of work that a submitter would need to do if they were to go one repository to another, but we do it all for them. So moving into the metadata that can be consumed, this is the EVA website. If you look at the top part of it, there's a menu with some instructions on how to submit data, then a study browser to search through the studies that are available in the archive, a variant browser to explore variants independently, a clinical browser for variants with clinically relevant information, some GA4GH demonstration of our API, documentation for said API, and then some help and feedback. So in the study browser, we list all the studies that have been already archived. This means that the metadata has been curated and all the correctness has been ensured. So by default, all the studies are displayed, and on the left side you can see that we allow to filter by different criteria. So one is the type of variants that that have been sequenced, so could be short genetic variants or structural ones. Also, the reference genome the study was sequenced against, and also the type of study. So, if we select one specific study, it is possible to see all this metadata information. As you can see, the first fields are related to taxonomy information. What is the reference genome? What are the species each sample belongs to? Because it could be the case that it's been referenced against human, but it's different primates or similar studies. The taxonomy ID, which links to the information in ENA. Then some information about the submitters, about the methods that were followed, the technologies, the number of samples available, and finally some links to the site where the project is hosted mainly, the FTP download, and the associated publications. The view for structural variants is similar. These variants are actually archived in a sister project called the Database for Genomic Variants Archive, which is dedicated only to structural variation and works in a collaboration with dbVar. The behavior and appearance of the browser for structural variants is mostly the same, the filters on the left side and information about the species and type of study on the table on the right. The view has a bit different metadata because of the nature of these two archives. We're trying to put them together as much as possible, and I'll mention this in a bit more detail in the future work. But it is pretty similar. So again, the different organism information, the methodologies, and some links to the publications and external resources. But in addition to the study browser, we also have the variant browser, which allows to search with a bit more granularity on the variant information that we have archived. So we recently had our first submission against the GRCH 38 assembly, the Illumina Platinum calls, and I wanted to show you how this would look in this browser. So on the left side, again, there are a set of filters. So we allow to search by genomic region, ensemble gene symbol, consequence types, frequencies, etc. The filter variants are displayed on the top right of the page, and you can see the most basic information, the location of the variant, the alleles involved, and also the consequence type and the polyvalent SIF scores extracted using VEP, the variant effect predictor by ensemble. More detailed annotation is displayed on the bottom right, where you can see also the gene IDs and symbols and codons if there were any changes, not in this case. And in addition to this, we also display in these other tabs information extracted directly from the VCF file, like the info columns or the header, which could also display the algorithms or tools that were run in the pipelines to generate the VCF file, a list of the sample genotypes, and also population statistics. So for instance, for 1,000 genomes, we have the population statistics generated. All this information is generated by a backend. So the website is just a view of these results. This information can be consumed by anybody else. So another portal could consume this information for a species of interest. For instance, we are collaborating with Ensemble Parasites, so they made submissions for 2 different species, and they are then consuming the API. So we store their data, and then they display it on their own portal. So this API allows to consume both study metadata and variant data. All the views that I have just shown, the study browser and the variant browser, are based on this API, which generates a response in a JSON format, which can be consumed by any programming language, so will work for any kind of website. And we also have an implementation of the GA4GH variation API. And if you're interested in using the API, there's a full documentation in the website. But I would like to show an example of how this would look if you were to consume it directly. So this is an example of the metadata for the 1,000 genomes phase 1. So on top of it, you can see the different query options, which in this case is just the unique ID assigned during the submission process. On the bottom, you can see all the information that was in the study browser: the name of the study, the description, the taxonomy, the species information, the methods, the PubMed IDs, URL to the resource, etc. And finally, the plans we have for next year. So first thing, we want to implement a tool to export the metadata information from EVA into a VD2K DDI compatible format. So all this information will be searchable in the Omics Discovery Index portal. And this is a functionality, but without the best metadata that we can provide, this wouldn't be really useful. So we want to improve our synchronization with the ENA. We want to provide links to sequencing experiments. Right now, these links are on the ENA side, but we don't have them directly linked from the EVA side. So we want to include that. And also, given that the ENA contains the whole view of a study, it's more possible that people will perform updates on their side than in ours. So we want to synchronize better these updates. There is also potential for doing more integration between the EVA and this other resource for structural variations, the DGVA. As you saw, there's small differences in terminology or in the information that's available from each of them. So we want to ensure that we provide, that we use the same terminology and we provide similar information from both to improve usability and make the information less confusing from the end user. And finally, we want to improve the species representation, showing a more complete taxonomic hierarchy, especially for those studies that are more complex, that involve multiple species sequenced against a reference genome. So, well, this is the team and all those that have helped us on our way. So thank you, everyone. Thank you all for your time. If you want to visit our site, this is the URL. And if you find anything that doesn't work as expected, or you would like to suggest any new features to be added, you're welcome to open a ticket on our repositories or send an email to our help desk service. So thank you, everyone.
Thank you.